Our final speaker today is Paul Richardson of DuPont Crop Protection. Paul currently serves as the Global Analytical Manager Seed Treatment Enterprise at DuPont. He's worked in the pesticide analysis area for 30 years and has been associated with seed treatment testing for the past 6 years. In conjunction with method development, Paul partners with external laboratories supporting regional field development, regulatory studies, and product stewardship. He will be speaking about analytical tools for the analysis of pesticide actives on seeds. Paul has his PhD in analytical chemistry from University of Georgia. Paul?
Thank you, Julie. I think I'm unmuted, so we can get started with the analytical part. And I think Amanda's last few slides, I think, were a good introduction to my part. In this talk, I'm going to talk a little bit more around the more common tools that we use to evaluate, to verify quality of treated seeds. This is an overview slide that's actually touching upon other attributes, again, testing and evaluations that Amanda was speaking of. So again, my focus is more around verifying the levels of pesticide that are on the seeds and then some related applications as well. Around active loadings, generally I think the industry, the people who are involved in this type of developmental work are usually taking a close look at pesticide loadings really from the beginning. It's something that we in DuPont keep a close eye on really during our early stage field trial studies. We want to make sure that there is confidence in what the conclusions from any biological testing is relative to what we know about the pesticide loadings on seeds. And then this just carries forward through all the way through commercialization to a point where we're interested in verifying that treatment equipment and our applications, technology, processes are delivering high quality and consistent treatments. Around pesticides, depending upon if we're talking about insecticides or fungicides, those are the more common areas that are examined. There's a pretty broad range of compositions that can be on seeds. Nonetheless, the most common tool that's used, I think, in a majority of places is LCUV. There are other tools, though, that have applicability toward pesticide testing on seeds, including LC mass spec, XRF, ELISA, and NIR-IR. Around the testing specifically around HPLC, you see the picture on the right-hand side. You're going to be extracting a known number or weight of seeds. There could be some optional milling prior to running extractions depending upon an active that you might be looking at. Some work has shown that there might be minor improvements in extraction efficiency depending upon how a seed subsample might be pre-processed before extraction. Filtration, centrifugation, something to lead toward a test solution that can be injected onto an HPLC. Generally, the HPLC separations are robust, but depending upon the workflow of a lab and the type of questions that are being answered, the chromatographic sequences can be somewhat lengthy. And as such, then some of the newer tools like UPLC has really been shown to offer significant improvements in separation speed and throughput in the laboratories. I was just going to speak very briefly about the separations part of this. There are scores of actives that are relevant for pesticide testing, and even in our own lab, we've encountered 40 plus actives depending upon what we're looking at, and we have found that this whole matter of managing separations has been greatly simplified by using applications that are available that can facilitate chromatography modeling. Through those kind of applications, number one, you can put in place separations of very complex mixtures, and the the approach is very quick and simple, but of more interest to us is the fact that this provides then kind of an evergreen database where we've captured a lot of retention behavior for many pesticides, and it gives us the flexibility to accommodate new actives where you might have different treatment partners on seeds. Not necessarily because the intent is to analyze any active that's on the seed, but it's just a way of assuring that you have a separation scheme where you know that you will not be prone to any interferences when testing the actives of interest. So this slide is just kind of highlighting that, and typical of this type of application, there's real-time feedback that can really expedite separations development, where the user can just modify potential gradient proposals and see very quickly where peaks of interest are going to be eluding. So I can't say enough about this particular tool, and again, in light of testing for many actives, we have found this to be a very convenient way of managing our separations lab, and basically we can handle anything on maybe a choice of one or two different chromatographic platforms. This right here is a true chromatogram, where on the previous slide that was a predicted chromatogram there. So in this particular case here, this was at a time when we were going to migrate to UPLC, and so we were screening many actives, and our intent was to have perhaps two chromatographic columns, a C18 and a phenyl column, and between a choice of those two columns and two different mobile phase systems, the intent was to be poised to address any analysis that might come our way. This separation here was nice in that it was a very rapid separation with a runtime of 3.5 minutes, really giving the chromatograph plenty of time to allow anything else elute that might be coming off later. We like the UPLC separations because it positions us to do sample-intensive tests, such as uniformity of distribution, which I'm showing on this slide here. This is showing what would happen when looking at single seed extractions and analyzing each extraction individually on the LC. By, in this case, measuring 50 seeds of coming from one application technique and 49 or 50 seeds for another one, and using the UPLC, we were able to generate these histograms that give a lot of information on what the consistency is in active loading across the individual seeds of a particular batch. And in this particular case, the technique associated with treatment B was one that yielded far higher, far tighter active ingredient loadings, and you can see an RSD of around 5.7% versus 13% in Technique A. So this provides very valuable feedback for formulations and applications technology, and the main point is, in addition to maybe just running a whole batch analysis where you see, on average, if your treatment is near or at target rate, this is a way of really understanding the consistency in a batch, and there are some attributes of the treatment process itself that can really have a major impact upon this. So just another area of quality that is monitored just through simple HPLC-based loadings analysis. There's also a lot of interest in pesticide measurements off seeds, where we might be looking at the release of pesticide into an aqueous media, monitoring its migration in the soil in and around a treated seed, monitoring its uptake into seedlings at various stages of seedling age. These type of analyses are where we reach out to an LC mass spec based testing. When looking at release in aqueous media, typically we might be looking at test solutions that might be around 25 nanograms per milliliter, but then when looking at soil and tissue, we're looking at a lower concentration of around 5 nanograms per gram. having this capability has proven useful because it allows us to answer certain questions, and one that's always of interest, and kind of going back to what Amanda was saying earlier, as different treatment recipes are being evaluated. You may be fighting multiple battles. You may want to control dust, but you also want to ensure that the active ingredient is migrating as you would hope, so that you're going to have the degree of protection that you have. So the off-seed measurements can help you to understand, how tightly the active might be held within a specific coating, as well as, what conditions or treatment formulas or recipes would influence the bioavailability. An example of testing active release in the water is shown on this slide. It was just a simple matter of understanding how tightly the active would be held by different coatings that were involving different coating ingredients, and a simple time release experiment like this can give you a sense of the rate of release per unit time. And this slide just simply shows a comparison of 3 unique treatments where there are agents on the seeds impacting release, and the trace at the bottom is an example of a treatment where the active was being held more tightly on the coating versus the trace near the top. So just some quick qualitative information, if you will, to get a sense of how different treatment components might impact the release of the pesticide. Tissue analysis is really helpful. It gives us an opportunity to measure the amount of active ingredient drawn in to the plant tissue. It's accommodated using the traditional conventional LC mass spec based residue methods. It gives us a chance to more carefully assess bioefficacy relative to what we know to be the true active ingredient concentration in the leaf, as shown in the little plot on the bottom. Another interesting aspect of tissue analysis is when you're carrying out a real-world field trial, maybe your insect shows up, maybe you have insect pressure, maybe you don't, and the cool thing is if someone bothers to take samples out in the field, you can nonetheless, under true field conditions, understand how well the active ingredient was being drawn up into the seedling. Moving on to commercialization, as we roll out a new treatment into a seed treatment manufacturing facility, there's a propensity for a lot of samples to build up, particularly when you're trying to assure that everything is optimized, and you're trying to essentially transfer your knowledge in treatment to maybe a different equipment setup at a specific site. So we've worked hard on that aspect of support, and a non-LC based tool, this one, X-ray Florescence, is very helpful depending upon the attributes of the pesticide of interest. In the case of this particular pesticide, there is a bromine atom on the molecule, and that bromine atom allows the use of X-ray fluorescence to measure its presence in samples, and that can then be used in order to correlate X-ray fluorescence signals to true loadings. We haven't found matrix interferences surrounding bromine, and we've seen excellent correlation with HPLC. This has applicability both to run batch analysis as well as to look at single seeds. And again, the intent is that, say if you are at a remote plant that does not have a traditional analytical lab, a portable XRF spectrometer could be used to measure the pesticides right there at line. The prep does involve some pre-processing. The seeds are milled and homogenized into fine powder, and then the fine powder is what is actually measured on an XRF spectrometer using a traditional sample cup. This is taken, around 4 to 6 minutes to complete once a sample arrives, so it's a very quick measurement. You do not have to wait for separation or for the finite time required for an extraction. And in a real-world example, this is where XRF measurements for pesticide were being used at the time that the unit was being scaled up for that treatment. And during the course of I forget how long the time frame was for the treatments, but there was time to gauge how things were going, and then there was an opportunity to evaluate pump calibration as it turned out, and then you can see through that feedback subsequent treatment loadings were in fact in alignment with the target. I mentioned a moment ago that XRF could also be used for looking at seeds on just a seed-to-seed basis. This is just a simple demonstration of how that would work. The illustration on the right is sort of showing what would happen at least with a small, somewhat round seed being interrogated from underneath by the XRF beam. And what you can do is you can look at the fluorescence emission from the coating on the seed, and it will correlate with subsequent HPLC measurements. So this particular series of plots are just showing how the XRF signal in bromine intensity correlates with subsequent HPLC testing with one replicate measurement, but then as soon as multiple replicate measurements are taken, you can see that there's a far higher correlation. It's not quite as easy with the larger seeds, but with the larger seeds there are other things that can be used that might allow there to be greater alignment between XRF measurements and the actual loadings on seeds. In this particular case, there were subsequent mass adjustments, just knowledge of the size of the seed, and knowing that you've characterized the coating on the seed, that would be a way of further aligning the data from the XRF to an HPLC. So these are just a few examples of some analytical tools that are specific for pesticides analysis on treated seeds, and that's what I have for you today. So thank you for your attention.